Okay, so this is going to be, we're going to talk about bacteria and the other prokaryotic group that's closely related to bacteria, archaea, or archaea, I should say. Okay, our learning outcomes, we're going to list the structures all bacteria possess, identify 4 structures that some but not all bacteria possess, describe 3 major shapes of bacteria, describe the unusual shapes of some bacteria, provide at least 4 terms to describe bacterial arrangements. Bacteria, archaea versus eukaryotes. Bacteria and archaea have nuclear material that's free in the cytoplasm. They do not have a nucleus. Eukaryotes have a nucleus. Internal structures, bacteria and archaea have no membrane-bound organelles. So this is a pretty busy slide. Whoops. And it shows you just an example of all the possible structures a bacteria could have. They have a cell membrane like eukaryotes. They have cytoplasm like eukaryotes. The bacteria have a cell wall usually. In gram-positive, it's thicker than the gram-negative bacteria. If you remember, those are the 2 major groups. They have an S layer, which is made up of protein. So these other protective coatings. They can have a capsule, which is usually made of sugars, that provides them additional protection. This can protect them against phagocytosis and therefore our virulence factor. The gram-negative bacteria have a lipopolysaccharide membrane, which is unusual membrane, unique only to the gram-negative bacteria, and also has a poison in it, an endotoxin that they shed that is not easy to break down. And is very resistant and is non-specific, but it is toxic. Bacteria can have, some bacteria that move can have this appendage called a flagella for movement. They have a single circular chromosome that's tightly wound up in the cytoplasm, but not bound by a membrane. Some bacteria, among the gram-positive bacteria, can form what's called a endospore, which is we'll talk about, it's a very resistant structure. And the bacteria will basically go dormant and stay in a suspended state in the endospore until it's in a more favorable environment. The bacteria also have something else that's unique. They have extra pieces of DNA called plasmids, which we'll talk about is carrying virulence factors, and we'll talk about how those are used in DNA technology. Bacteria do have, keep saying I have some people to join and I don't see them. They do have, bacteria has ribosomes like our cytoplasm does. Their ribosomes, though, are a little different, a little simpler in structure. They also have these fimbriae that can enable them to attach to surfaces. Okay, sorry, I didn't get these people admitted. Okay, bacteria are single-cell organisms. They can live in colonies, they can reproduce in groups, but they are all single cellular. They carry out the basic functions necessary for life. They can reproduce, they can carry out metabolism, they can process nutrients. They can act as a group in a colony, but they are not multicellular. They can also form groups on these organized on these biofilms, which is
Professor.
basically what plaque is. It's a biofilm. It
Professor.
enables them to adhere to your teeth and is a resistant group opposed to the bacteria being by themselves. Bacteria size, they consider there's considerable variety in size, shape, and their colony arrangements. The average size of a bacteria is 1 micron. There's a very large bacteria that's 100 to 750 microns. It's unusual. There's also very small nanobacteria that's can be down to 0.2 microns. The 3 general shapes of bacteria are the coccus, which are round spears, oval shape. You have the bean-shaped bacteria. You have the bacillus, which is cylindrical. Filamentous bacteria that's kind of club-shaped. Vibrio, such as the bacteria that causes cholera, Vibrio cholera, looks like a little comma shape. Some of these shapes are in here. Some bacteria exhibit pleomorphism. Morphology refers to shape. Pleomorphism means they have a variable shape. So they're not always uniform. So these are some of the common shapes. I said the coccus was spherical. The bacillus are rod-shaped. There's some variation. When you see bacteria like this in a chain, they are not multicellular, but they stay joined together. You have various bacteria that have these curved shapes. Vibrio would be like this, looks like a little comma. Here's some Staphylococcus aureus that grows together in these groups. Here's the Vibrio, which has slight curve to it. The Streptococcus pyogenes causes strep fever. They stay together in these chains. And there's the Streptococcus bacillus. It forms these chains. They just, each time they reproduce, they just stay joined together. Mycoplasms are unique in bacteria. They're very small. They can cause pneumonia. They do not have a cell wall. So they have a lot of, not having that rigid cell wall. They have a lot of variable shapes. They also have something else unique to them. They have cholesterol or sterols in their cell membrane. Most bacteria do not have sterols in their cell membrane. Most eukaryotes like us, we have sterols and cholesterol in our cell membrane, helps to provide structure. Arrangements and grouping of the cocci, they are the spherical shape. There's Diplococcus. They're always in pairs. Tetrads are always in fours. The Staphylococcus and the Micrococcus are these irregular clusters. The Streptococcus are in chains of just a few to hundreds of cells. Sarcinia is a cube-shaped packet of 8, 16 or more cells. Just keep in mind, these are not multicellular. Possibly it was the origin of becoming multicellular if they're if they became differentiated in their function, but this is how they grow together in groups. Other bacterial arrangements of the bacillus. There's their diplobacilli. They're in pairs with the ends attached. The streptobacillus are long chains. A palisade arrangement is the cells of a chain that remain partially attached and fold back, creating a side-by-side row of cells. The spiralia are occasionally found in short chains, and spirochetes rarely remained attached after cell division. What are three common structures that all bacteria have? Anybody? Okay. Three common.
Cell membrane.
Yes, they all bacteria have a cell membrane that is common to them. What else?
Cytoplasm.
They have cytoplasm, and what would be a third common?
Ribosome.
They have ribosomes, that's correct. List and describe four structures found in at least some bacteria. What would be some structures that a lot of bacteria have, but not necessarily all of them? What about the cell wall? Do all bacteria have a cell wall?
No.
No, remember the Micrococcus, very small bacteria do not have a cell wall. Gram-positive bacteria have a very thick cell wall. Gram-negative have a thin cell wall. Another structure that's a lot of bacteria have, but only found in the gram-negative bacteria, is the lipopolysaccharide layer, which is an additional unique membrane found only in gram-negative bacteria. What is a unique structure you might see in some gram-positive bacteria? I'm thinking of the endospore is a structure that you might see in some bacteria. Three general shapes of bacteria. Do you remember what some common shapes are?
Rod.
Yeah, the rod bacillus shape. What are two other shapes?
Curve.
Pardon?
I said curve.
Curve, yeah, that little curved shape that the Vibrio have. And what's the third shape?
Cockles.
Oval.
Yes, that's the coccus is oval or spherical shape. What are two arrangements found among the general shapes of bacteria?
Staphylococcus.
Yeah, the Streptococcus, so those would be in chains. If we said Diplococcus, they would be in pairs. Also in bacilli, you can also have chains of arrangement of a chain of bacteria. Describe the structure and function of 6 different types of bacterial external structures we're going to talk about in this section, and talk about how the flagellum works. Not all bacteria have a flagellum. It's an appendage of motility. Two major types of appendages are the flagella, and the other ones are these axil filaments that some bacteria have. A filament wraps around the bacterial cell. The bacteria moves in a corkscrew-like fashion. The fimbriae act like little propellers that rotate. Also, bacteria have various structures for attachment. There's fimbriae, pili, and nanowires that can enable them to adhere. So the flagella anchors into the outer membrane. If it's a gram-negative bacteria, the cell wall all the way down into the cytoplasm, and the structure can rotate. The flagella looks like a thin, long, whip-like structure, and there's different arrangements of these flagella, but they enable the bacteria to move. Not all bacteria are mobile. So the different arrangements of the flagella, you can have a single flagellum, mono, single trichus means hair-like. So this would be a monotrichous bacterium. Lophotrichus has small bunches or tufts. So these would be bacteria here would be Lophotrichus. If they have flagella at both ends, they're called Amphotrichus. And if they have flagella, a little hard to see, that's dispersed randomly around the surface, they are Peritrichus. Chemotaxis and phototaxis. So this is, the bacteria can respond to chemicals in the environment and light, and they can move towards the chemical or the light, or they move away from it. So positive chemotaxis, the cell moves in the direction of the chemical stimulus. They have receptors that can sense various chemicals in the environment. Negative chemotaxis, they move away. Phototaxis, the photosynthetic bacteria can move toward the light. So run versus tumble. So the flagella rotates in a counterclockwise movement, and it can move in a straight line. If it senses something, wants to get away from, it rotates the other direction and starts to tumble. And once it's facing the other way, it can start moving away. And this just shows you this is a monotrichous bacteria and a paratrichus that has lots of flagella all over. And so this just shows you the sort of random zigzag movement the bacteria can make when it's going back and forth between a tumble and a run. And it's due to being either attracted to a chemical or moving away from a chemical. And if it's moving towards something, it's positive chemotaxis, and away, it's negative chemotaxis. Paraplasmic flagellum. So some bacteria have these axial filaments that actually wrap around the cell, like the spirochetes. And the bacteria then rotates in a corkscrew-like fashion to move, imparts a twisting or flexing motion of the cell. Attachments that bacteria have, structures for attachments, can also make them more pathogenic. An example is the pilus structure. It provides adhesion but not movement. There's also fimbriae, little structures for adhesion. In some species, even the flagella can be used for attachment. Fimbriae are these small bristle-like fibers on the surface of the bacteria. Composition varies, but most of them are just made of protein. They have a tendency to stick to each other and to surfaces. And it may be responsible for formation of biofilms. E. coli uses Fimbriae to adhere to epithelial cells. So that would be considered a virulence factor. Pili, and we're going to talk about more about these in the genetics part and in DNA technology. The pili are long, rigid, tubular structures of pilin protein. Only gram-negative bacteria have them. They're used in a process called conjugation. As you can see, some bacteria here being joined together by the pili. The bacteria can actually transfer DNA from one cell to another. There's a specific gene the bacteria has to have to make the pili and exchange the genetic material. They can have some other structures. So the S layer, this is outside the membrane, outside the cell wall. The S layer is a single protein joined together. It provides protection from environmental conditions. It's only produced when the bacteria is in a hostile environment. Another layer is called a glycocalyx, which is just repeating units of a sugar that may or may not include protein. The slime layer is formed loosely around a cell and protects them against the loss of water and nutrients. And another important structure is a capsule. It's more tightly bound than a slime layer. It's denser and thicker. It plays an important role in virulence of bacteria, can enable them to evade phagocytosis. We'll talk about capsules a bit more in DNA technology. The slime layer here, you see a little more disorganized. Capsules, a very organized structure, but both of them provide protection, additional protection. So when you do a stain, the capsule here shows up because it repels the dye, the background stains. You can also stain the bacterium. The other thing about capsule and virulence, you can see there's a difference when you grow these in colonies. So like Streptococcus can have a capsule or not have a capsule. So if they have a capsule, they make a very orderly, smooth colony. If they don't have a capsule, it's a more kind of disorganized looking colony. Capsules are formed by many pathogenic bacteria. They protect against phagocytic white blood cells. Biofilms, some examples of a biofilm, the plaque on teeth protects bacteria. They can infect long-term indwelling artificial devices through the formation of a biofilm. The biofilm is made up of bacteria and extracellular glycocalyx slime to hold the bacteria together. Biofilm formation starts first, some bacteria colonize the surface. They stick to the surface. As they divide, they start making the glycocalyx. Additional bacteria can become attached and create a mature community with complex function. So you can see why this is a big problem in dentistry. These biofilms, why you got to get the plaque off your teeth, because the bacteria can just keep growing and cause disease. So there were 3 parts to a bacteria flagellum. Let me go back and look at that real quick. It had a filament, which these long filaments stick out. They have a hook region, and they have a rod and rings that embeds it into the outer membrane, cell wall, and finally down into the cytoplasm. What's the difference between a run and a tumble with the flagellum? Do you remember the difference?
Was
So
it the direction of movement?
Correct. So when they are moving on a run, they're moving towards chemical in the environment. In a tumble, the flagella starts rotating the opposite direction, causing the bacteria to tumble. True or false? Bacteria fimbriae are used for conjugation. Is that true or false?
True.
That is false. The structure for conjugation is called apelli. Fimbriae are more like these small hair-like structures. What's the difference between a capsule and a biofilm? So if you think of the biofilm as like that glycocalyx, what was the difference? The capsules are more orderly in structure than the biofilm. The capsule is what surrounds an individual bacteria. Biofilm is a group of bacteria. The cell envelope is a boundary layer of bacteria. Differentiate between 2 types of bacteria envelope structure. Discuss why gram-positive cell walls are stronger than gram-negative. So what are the 2 major groups of bacteria that we talked about last week? When you do a gram stain, what are the 2 major groups?
Is it positive and negative?
Yeah, that's correct. So when you stain them, most bacteria will be either gram-positive, gram-negative. There are exceptions, the mycoplasms, the bacteria that cause tuberculosis. They have a thick, waxy layer. But most bacteria, gram-positive bacteria, have a thick cell wall. Gram-negative bacteria have a thin cell wall. The gram-negative bacteria have this additional structure called a lipopolysaccharide layer. And that lipopolysaccharide layer also has a virulence factor, a non-specific O protein in the lipopolysaccharide. Gram-positive cell walls are stronger because they are thicker. Name a substance in the envelope structure of some bacteria that can cause severe symptoms in humans. So I already mentioned that. That is endotoxin. Endotoxin is non-specific, and it's a resistant structure, and it is a virulence factor. The cell envelope is basically 2 to 3 basic layers. The cell wall, the cytoplasmic membrane that most bacteria have, and the outer membrane, the lipopolysaccharide layer that gram-negative bacteria. Together, these either 2 or 3 layers act as a single protective unit in the bacteria. The Gram stain, this is important to know, goes all the way back to 1840. It delineated two major groups of bacteria, lies in that structure, that cell envelope. The Gram-positive bacteria, the thick cell wall. Cell walls in both instances are made of peptoglycan. Bacteria, of course, has an inner cytoplasmic membrane. The Gram-negative have an outer membrane, the lipopolysaccharide layer. They have a thin They have a thin cell wall, and they also have a cytoplasmic membrane. Comparisons of the two. So both Gram-negative, Gram-positive bacteria have a lipopolysaccharide layer. Have a cell membrane. Cell membrane is a phospholipid bilayer. There's common structure of cell membranes we have, and a lot of other eukaryotes have also. You can have proteins that are embedded in or on the cell membrane. You can have enzymes, various structures that enable things to get in or out of the cell. In bacteria, in the Gram-positive bacteria, they have that thick cell wall. They also have this structure called teichoic acids that attaches the cell membrane and spans the entire cell wall. Viruses that infect the bacteria can bind to these teichoic acids. The Gram-negative bacteria have a cell membrane, thin cell wall. They have the lipopolysaccharide layer, which also looks a bit like the phospholipid bilayer that we see here. It has these channels in it for things to move out. It has lipoproteins, proteins that have lipids to them. These large structures are called porins that can allow things to go in or out of the cell. In the bacteria, the lipopolysaccharide layer has these either sugars or proteins that have lipids conjugated to them. By the
What
way,
is the
when you
difference between them?
the difference is both bacteria, though they have a cell wall in Gram-positive, the cell wall is much thicker. The cell membrane is very similar, but another difference is what we call the lipopolysaccharide layer that's only in Gram-negative bacteria. So only the Gram-negative bacteria have it, and the lipopolysaccharide layer has porin channels and either phospho or lipoproteins that are unique to them. So when you stain bacteria, do the Gram stain, what color does the Gram-positive bacteria stain?
Purple,
Purple. And the Gram-negative stain what?
pink.
Pink.
Yeah, pink. Yeah, Gram-negative. And most bacteria are either Gram-positive, Gram-negative. Gram-positive bacteria are the ones that can form endospores, and the Gram-negative are more resistant to antimicrobial treatments because of this lipopolysaccharide layer. Structure of the cell wall, it helps determine the shape of the bacterium, provides a strong structural support to keep the cell from bursting or collapsing due to osmotic pressure. The peptoglycan layer is found in the cell walls of most bacteria. That's what the cell wall is composed of. which is a sugar with short peptides, which are short little chains of amino acids. It is unique macromolecule. So I was saying these glycans, which are sugars that are cross-linked with short peptide fragments, which are amino acids. It provides a strong but flexible support framework for the bacteria. The structure of the peptoglycan in the cell wall. This just shows you it's just these two sugars and acetylglutamic acid, which is G, and muromanic. I think that's what it, if I remember right. Yeah, muromanic acid. And acetyl stands for a glucosamine, which is a sugar with an amino acid hooked to it. And you can see that the short little chain of peptides, which are amino acids, help to cross-link. So you have one chain here, then another chain, another chain, another chain, and amino acids help to hold the chains together. The gram-positive cell wall is thick, homogeneous, about 20 to 80 nanometers thick. It has some teichoic and lipoteichoic acids span the cell wall. They function in cell wall maintenance, enlargement during cell division of the bacteria. They have an acidic charge on the cell surface of the bacteria. Remember, bacteria are generally negatively charged, so dyes are usually positive. What about if you do a negative stain? Is the dye negative or positive? The negative, it is negative, so it's repelled. So the background, when you do a negative stain, the background stains, but not the bacteria. Gram-negative cell wall is thinner than the gram-positive. It's a single thin sheet, 1 to 3 nanometers. The gram-positive is 20 to 80. It is a somewhat rigid structure. Its thinness gives gram-negative bacteria greater flexibility, but makes them more sensitive to lysis. To lysis something is to break it open. Some non-typical cell walls. Some bacteria lack a cell wall structure, and they are that we see in gram-positive or gram-negative. The bulk of cell walls are microbacterium, and the Nocardia is composed of unique lipids, such as macolic acid, or something called cord factor, a very long chain of fatty acid. It contributes to the pathogeneity. So microbacterium cause tuberculosis and leprosy. It makes them more resistant to chemicals, dyes, and antibiotics. Instead of using the gram stain to identify these bacteria, you have to use the acid-fast stain to identify them. Mycoplasms, the very small bacteria, remember we said they do not have a cell wall. They have sterols in their cell membrane, which helps to protect them from lysis. They're pleomorphic, that means they have variable shape. They can range from 0.1 to 0.5 microns. They can have a filamentous to a coccus shape or even a donut shape. Cytoplasmic membrane is about 5 to 10 nanometers. It's a flexible sheet hold it completely around the cytoplasm. The cell membrane is a phospholipid bilayer embedded with proteins. It's composed of about 30 to 40% phospholipids, 60 to 70% proteins. Bacteria, the mycoplasms, have high amounts of sterols. Archaea contain unique branch hydrocarbons rather than fatty acids in their cell membrane. The function of the cytoplasmic membrane, energy reactions occur on the cytoplasmic membrane. It's involved in processing nutrients and synthesis. It regulates transport of nutrients into the cell and the removal of waste from the cell, discharging the waste. Cytoplasmic membranes are selectively permeable. Water and small uncharged molecules can freely diffuse through the cell membrane. Other than that, you need special carrier mechanisms for passage of most molecules. Secretion is the discharge of metabolic products into the extracellular environment. The gram-negative outer membrane has specialized polysaccharides and proteins. So it has that lipopolysaccharide layer. It has the polysaccharides can function as cell markers and receptors. I mentioned before they have an endotoxin in the lipopolysaccharide layer. The endotoxin is a non-specific toxin. It can cause fever and shock. It's very resistant. The lipoproteins help anchor the outer membrane to the peptoglycan in gram-negative bacteria. I pointed out these large porin proteins that span the outer membrane. They only allow relatively small molecules to penetrate. The size can be altered to block the entrance of harmful chemicals, and it can even act as a defense against certain antibiotics. Now, the gram stain is important to know, and these are the steps for the gram stain. So first, I'm sorry about that, I keep hitting that. So if you're doing a gram stain, the first thing you do is you stain, let's say you just had a colony of bacteria, or let's say a mixture of bacteria, and you had a mixture of gram positive and gram negative. So first you would stain with the crystal violet, which is stained purple. You then have to use an iodine, acts as a mordant. This causes the dye to fix in place if it's gram positive. You then do an alcohol wash. So the alcohol wash dissolves the lipids of the outer membrane, which is what the gram negative bacteria have, found only in the gram negative cells. And so then is your last step, you stain with saffron. Oh, it's a red or pink dye. It gives a red or pink reaction. Gram positive bacteria won't stain because the dye from the first step, the crystal violet's already fixed in there. Since the lipopolysaccharide layer has been washed away, now the dye, the saffron, can stain the cell. So again, in the first step, both cells affix the dye, the crystal violet. When you add the iodine, it becomes complex. The dye complex traps into the cell wall. The iodine has no effect on gram negative. When you do the alcohol wash, the crystals remain in the cell wall, gram positive. but the outer membrane is weakened and the cell wall loses the dye if it's gram negative. Some differences in the cell envelope, outer membrane and gram negative bacteria makes them resistant to some antimicrobials. They are more difficult to inhibit or kill than gram positive. Infections with gram positive bacteria are treated differently than infections with gram negative bacteria. The cell envelope in disease, the cell envelope can interact with human tissues and contribute to disease. Proteins in the outer cell wall of gram-positive bacteria can be toxic. Lipids in the cell wall of Mycobacterium can be harmful to human cells. Large molecules in the cell wall are foreign to humans and can stimulate an antibody production. Okay, what are three differences between gram-positive and gram-negative bacteria? Do you remember the three differences between the gram-positive, gram-negative bacteria?
Cell walls.
And
The
what were these?
gram-positive wall is thicker.
Yes, what's another difference? So the gram-negative, what structure do gram-negative bacteria have that gram-positive don't have?
Thick membrane.
They have that lipopolysaccharide membrane on the outside, and their cell wall is thinner than the gram-positive bacteria. The outer membrane of gram-negative bacteria, how does it contribute to disease? It has an endotoxin in it. So throughout this class, we'll talk about virulence factors. And endotoxin is a virulence factor. It is non-specific. It is a resistant toxin, not easily broken down the way other toxins are. The cytoplasmic membrane is selectively permeable. Simple things that aren't charged can move through the cell membrane. Others have to be specially transported. Okay, so let's take, it is about 5 after 8. Let's take a short break until 8:10, 5-minute break. We'll start back 10 minutes. I am recording this lecture, and I will either put a link in an announcement, or I'll try to post the link in the modules. I can't post the actual video in the modules. They don't, the servers don't have enough room for that. It's actually at the end of the lecture, I have to convert this, and it's actually takes a pretty long time. So we'll start back at 10 after 8. Okay, so let's start back. Okay, we left off here. We're going to talk about bacterial internal structures. Identify 5 structures that may be contained in a bacterial cytoplasm. Detail the causes and mechanisms of sporulation and germination. So only the gram-positive bacteria can form spores. The cytoplasm is a gelatinous solution contained by the cytoplasmic membrane. It is a prominent site for cells' biochemical and synthetic activities. The cytoplasm is 70 to 80% water. It has a mixture of sugars, amino acids, and salts. It has the chromatin body, in other words, the DNA. It has ribosomes. It has granules and fibers that act as a cytoskeleton. So the cell actually has an internal structure. Somebody said they had to log in and log out. That's no problem. I'll just have to make sure that I see you. But I do get, well, I say that, but I get each time somebody logs in, I get a message saying to admit somebody. So do what you have to do. So bacterial chromosome. So bacteria have a single circular strand of DNA. We have linear chromosome. We have paired chromosome. The bacterial chromosome aggregates in a region of the cytoplasm called the nucleoid, but this is not a nuclear nucleus. It's not membrane bound. The DNA is tightly coiled around basic protein molecules to fit into the small compartment. Bacteria have a unique form of DNA that's outside of the chromosome. It is called plasmids. These are non-essential. So the bacteria does not need a plasmid to grow and reproduce.
Are these
These-
like small DNA?
Yes, small pieces of DNA. There are separate double-stranded little circles of DNA, but they play an important role in virulence. They can be duplicated and passed on to other bacteria during replication. They can carry protective traits for the bacteria. And they're widely used in genetic engineering because you can take a plasmid, cut it, put a gene in it, insert it into a bacteria, and the bacteria will make the gene products. Ribosomes, we'll talk about this in our next section about molecular genetics. The ribosomes are made of 2 things. They're made of RNA and protein. They have 2 subunits, a large subunit, 50S, and a 30S. Together, when these are joined together, it's a 70S. These aren't additive. The S refers to a sedimentation factor, not to molecular weight. But this is the site of protein synthesis, which we'll talk about in our genetics section. The bacterial ribosomes are 70S, and eukaryote, it's 80S. Bacteria can have inclusion bodies. These are storage sites for nutrients. They vary in size, number, and in the content. The cytoskeleton of bacteria is proteins made of actin, which is similar to eukaryotes. They are arranged in helical ribbons around the cell, just under the cytoplasmic membrane. They help contribute to shape. They can also be a target for antibiotics. Now, Endospores is another bacterial structure, but only gram-positive bacteria have them. The bacteria goes into a dormant state. The endospores can withstand hostile conditions and facilitate survival. The endospores are part of a two-phase life cycle. So when the bacteria is normally growing, we call it a vegetative cell. That refers to just growing and reproducing. It's not a vegetable, but it's metabolically active. When it's an endospore, it's inert, resting condition. When the endospore comes into a favorable environment, sporulation happens and the spore gives rise to a new bacterial cell. Endospores are very resistant. They are a virulence factor. They can resist heat, drying, freezing, radiation, and chemicals. But the gram-positive bacteria that make spores, being in an environment with a lack of nutrients, especially carbon and nitrogen, they'll start making a spore called a sporangium, sporulating cell. It takes about 6 to 8 hours for the bacteria to make a spore. So what happens in an unfavorable environment is the chromosome is duplicated and separated. They start forming this structure, so it separates and gives rise to a spore. You'll initially see the spore inside the bacterial cell. And what happens is, as the spore matures, the rest of the cell breaks down and the endospore is released. And then when what's left, if this comes into a favorable nutrient-rich environment, this spore germinates and releases a new bacterial cell. Germination begins with favorable conditions, such as water. Germination agent stimulates the formation of hydrolytic enzymes that break down the cortex. So the cortex is helping protect the spore there. The core rehydrates, it takes up nutrients, and the bacteria grows out of the endospore. Once this process starts, it takes about 1 to 5 hours. So it takes about 6 to 8 hours to make a spore and about 1.5 hours to make a new bacterial cell. Diseases related to spore-forming bacteria. Keep in mind, these are all gram-positive bacteria. You've probably heard of these. Bacillus anthrax causes anthrax. Clostridium tetninum has a neurotoxin that it secretes. It causes tetanus or paralysis. There's Clostridium perfringens that causes gas gangrene and badly infected wounds. And then there's Clostridium botulinum, which can cause very severe food poisoning that is fatal. It releases a botulinum toxin that is a neurotoxin also. Endospores are constant intruders where sterility and cleanliness are important. They resist ordinary cleaning methods. Boiling water, soaps, and disinfectants won't stop them. They can frequently contaminate cultures and media. Hospitals must protect against endospores and wounds. The destruction of endospores is important in the food canning industry. So true or false, plasmids, are they vital to the metabolism of bacterial cells?
False.
True.
False.
Mhm.
You're saying
False.
true or false?
False.
False, yeah. The plasmids are not essential to survival of a bacterial cell. Plasmids, however, can carry variance factors such as resistance to antibiotics and resistance to heavy metals. So how are bacterial ribosomes different than eukaryote ribosomes? They form a 70S unit and eukaryotes 80, but they actually have fewer types of proteins in them. So they're a little simpler in structure. So the steps of endospore formation, probably easier just to go back and look at it. So the first step, you duplicate the genetic material. You form this sporangium that gives rise to the spore. Eventually, the genetic material is walled off. The other genetic material eventually breaks down and you have a spore. And it has various layers, a core, cortex, endospore, exospore. And when the spore gets into a favorable nutrient-rich environment, the spore itself winds up releasing a new bacterial cell. What are 3 diseases caused by endospore forming bacteria?
Tetanus.
Tetanus is one. What are 2 other ones? Botulinum, and what was the other one?
Clostridium.
Anthrax.
Clostridium.
Yes, Clostridium botulinum causes, so both botulism and tetanus is due to a neurotoxin that the bacteria secrete. And then there's anthrax, which is caused by a gram-positive bacillus. So we'll talk a little bit about the archaea and some of the differences. They are both prokaryotes, but some of the differences between them and bacteria. Archaea are more closely related to eukaryotes than they are to bacteria. They have ribosomal RNA sequences not found in bacteria. Their protein synthesis and ribosomal subunit structures are similar. They have genetic sequences found only in the ribosomal RNA. They have a unique method of DNA compaction. They have unique membrane lipids, cell wall composition, and pilin proteins. Comparison of the 3 domains. So we have bacteria, archaea, and eukaryotes. So the chromosomes, both bacteria, archaea, they have a single circular chromosome. Eukaryotes usually have paired linear chromosomes. There are ribosomes, are similar, but the structure, even though this is a 70S, its structure is more similar to the eukaryote. Their flagella are different. They have hook rings and a hollow filament. Archaea have a solid fimbrial-like structure. Eukaryotes that have flagella, single-cell protists that have flagella, they have what's called a 9-2 microtubular arrangement. They all have unique ribosomal sequences that only identifies them. So if you sequenced the ribosomal RNA, you would see that is for bacteria, it's unique and different than the archaea, eukaryotes, and so on. So these all have unique sequences. Protein synthesis is similar between the archaea and eukaryotes, but different in the bacteria. Archaea don't have peptoglycan in the cell wall, neither do eukaryotes. The cell membrane lipids are fatty acids with ester linkage. Archaea, they have long chain branched hydrocarbons. And actually the eukaryotes also a little more similar to a bacteria. Sterols in the cell membrane. Archaea don't have it. There are some, like the mycoplasma bacteria, have it, and we have sterols in our cell membrane. Characteristics of archaea, they are the most primitive of all life forms. They are closely related to cells that originated 4 billion years ago. They live in habitats that are similar to the extremes found in the ancient Earth. There was heat, salt, acid, pH, pressure, and the atmosphere was different. They are methane producers. They are what we call hypothermophiles. They love really hot temperatures. Or extreme halophiles, means they like very salty environments, and they can reduce sulfur. So the archaea are methanogens. They can take CO2 and hydrogen gas and make methane. They are common inhabitants of an anaerobic swamp mud, bottom sediments of lakes and oceans. They're also found in the digestive tracts of some animals. The gas produced in swamps has become a source of fuel. They may contribute, though, unfortunately, to greenhouse gases in global warming. Extreme halophiles, they actually require salt. They can multiply in 36% solution of sodium chloride that would destroy most cells. They exist in inland seas, salt lakes, salt mines, in fish that have been salted. They use a red pigment to make ATP in the presence of light. So these are some examples, halophiles from around the world. Now, some bacteria are psychrophiles. And what that means is they like to grow at very low temperatures. Hypothermophiles grow at very high temperatures. So a hypothermophile, they would flourish at 80 to 113 Celsius. They cannot grow at lower temperatures. They live in volcanic waters, soils, and submarine vents in the ocean. They are often tolerant to high salt and acid as well. How are archaea cell walls different than bacteria cell walls? So if you go back and look at the chart, they do not have peptoglycan in their cell wall like the prokaryote bacteria do. Well, they're basically prokaryotes too, but they do not have peptoglycans. So define the terms. What's a methanogen? It is an organism that makes methane. So the archaea can make methane gas. Some of them are halophiles. What's a halophile? They love a high salt concentration. Psychrophiles like very cold temperatures, and hyperthermophile like very hot temperatures above 80 Celsius, above 50 Celsius. Do the archaea cause human disease? As far as we know, they do not. Let's see how many slides we have left. Okay, so I'm going to talk about classification and the manuals used. There's Bergey's Manual of Systematic Bacteriology and Bergey's Manual of Determinative Bacteriology. We're also going to talk about 4 divisions of bacteria ending in the suffix cutes and describe their characteristic, and define a species in terms of bacteria. Classifications. Systems have both a practical and an academic purpose in differentiating and identifying unknown species and studying the relationship and origins of microbes. The evolutionary relationships that can trace them. Many bacteriologists classify according to shape, arrangement, and growth. Newer methods, we rely on biochemistry, genetics, and molecular traits to classify on them. Evolutionary relatedness is most accurately determined by sequencing the ribosomal RNA of bacteria. It tends to remain stable in the nucleic acid content over long periods. Any major differences in signature sequence indicate a distance in ancestry. The Bergey's Manual is the definitive published source for bacteria and archaea classification. Phenotypic traits are the traits that we can see and study. They're a basis for the early classification. Traits such as shape or how they behave in culture and biochemical reactions. It is still used by clinical microbiologists and researchers who need to quickly identify an unknown bacteria. So they would look at their shape, they would stain them, look at what kind of biochemical reactions they can carry out. The Bergey's Manual of Systematic Bacteriology relies on phenotypic information, but also RNA, the ribosomal RNA sequencing. It's now available online. The Manual of Determinative Bacteriology is based entirely on phenotypic characteristics. So these are things that would be assayed in a clinical, teaching, or research lab. It is still widely used by microbiologists who need to identify bacteria but don't need to know their evolutionary backgrounds. It is useful for students of medical microbiology. So some major divisions of bacteria in Archaea. The gracilicutes are gram-negative bacteria. So remember, they have the thin cell wall. Firmicutes are gram-positive bacteria. They have the thick cell wall. Tenricutes lack a cell wall and are soft, so those would be like the mycoplasms. And the Archaea are Mendosicutes, I believe. Those are the 4 major groups of all bacteria. Diagnostic scheme. So you would first determine whether something was either gram-negative or gram-positive, and whether it had a cell wall. You would look at shapes, arrangements, and oxygen. So bacteria, most bacteria are aerobic, but some are anaerobic. In other words, they don't use oxygen in their metabolisms. Some of them are facultative. They can switch back and forth between being aerobic and anaerobic. Medically important families and genre of bacteria. So I'm not going to go through all of these. You can look at these yourself. I don't believe the test has requires you to know specific bacteria. And these are common bacteria, gram-negative bacteria that are important medically. Medically important families and genre of gram-negative bacteria and no cell wall, such as the mycoplasma and a related ureaplasma that causes urinary infections. Species and subspecies of bacteria in Archaea. Bacterial species, a collection of bacterial cells which share an overall pattern of similar traits. A subspecies, strain, or type. Bacteria of the same species that have differing characteristics. A serotype is a species that simulates a distinct pattern of antibody response because of distinct surface molecules. What part of the bacterial cell is analyzed to determine evolutionary relatedness? So that would be the ribosomal RNA. The Burgess manual is often used in a clinical laboratory. If you remember, it's the determinative. Bacteriology used in clinical situations. The definition of species. How's the definition of species different for bacteria? It's just differences in characteristics, and a serotype is determined by its reaction to antibiotics. Okay, so that's the end of that. I'm going to stop sharing this for a second. It doesn't, I don't think it turns off the video till I end the lesson. So does anybody have any questions at this point? We have one more lecture next week, February the 15th. I'm sorry, February the 8th, and in 2 weeks on February the 15th, we have exam 1. So are there any questions before we go? Okay, so I'm going to end.
No, I don't have a question.
Okay, go ahead.
No, I said I don't have a question.
Oh, okay, sorry. So if there's no questions, I'm going to end this and hopefully be able to post the video.
Okay.
All right, so good night,
I have a
everybody.
question.
Yeah, go ahead.
I have a question.
Yes,
Can you hear me?
yes, I can hear you. Go ahead.
Um,
Brenda's talking.
Where can we find the
I'm sorry, you're
recordings?
breaking up a little.
She said, where do we find the recordings, Professor?
So I'm going to either, like I posted one just as a test in an announcement. So it'll either be an announcement or I'll try to it'll have to be a link because I can't post the whole video. So I can only post a link to it. And it'll either be on a announcement or I'll try to put the link in the module. And I'll let you know which one it is.
Okay.
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 All right. Good night, everybody.
Good night, Professor.
Good night, Professor.
Night.
Good night, Prof.
Have a good
Good
week.
night.
Good night.